Hi guys, we're back with the second video here. Second video is going to take us through the stages with which our bodies can actually create these gametes, which I talked about in the first video. Okay, and so we are going to get into a deeper talk here about meiosis and how meiosis reduces the number of chromosomes in a normal cell from diploid to haploid in the gametes. Okay. Now, one of the things that you'll notice here, and some of this will help you, but at other times it's going to hinder you, is that this process of meiosis is similar to mitosis. Okay. In a couple of different ways. Number one, it's preceded by the replication of chromosomes. So we have to understand that before meiosis ever happens, the chromosomes do replicate, and that same process, that same event happens in mitosis as well. Not only that, but we will also see some of the same events, and some of the same events in meiosis are actually named exactly the same as mitosis. So we'll see separation of sister chromatids. We'll see separation of genetic material, if you will. Genetic material will be packaged into separate cells at the end. But as far as the similarities, that's really about it. There are substantial differences from mitosis. Okay. Now, first off, in meiosis, there are two sets of cell divisions instead of just the one in mitosis. At the end of meiosis, there are 4 daughter cells as opposed to just 2 in mitosis. And finally, and maybe most importantly, each daughter cell in meiosis is haploid. Not only that, it should technically be genetically distinct, and we'll talk about how that happens. We'll the final video discuss how that happens a little bit more in depth. Okay, so there are some substantial differences between these two types of things, although there are some similarities that will help you learn the events of meiosis a little bit more quickly. Okay. Now the College Board is going to, there we go. The College Board is going to want you to know a lot of these similarities and differences. So do keep your eyes peeled for those and your ears open, so that when they do pop up, you make note of them and maybe write them in your notebook, or if you have this PowerPoint slides up, make sure you put the little note in a specific slide. Okay. All right, so that's a majority of what I'm gonna make sure that I get across to you today, is the fact that the College Board does look for they want you to know the similarities and the differences, and they want you to know how those differences affect the ultimate outcome of meiosis. Relatively little detail is taken into account when the College Board wants you to know about the specific stages of meiosis, so like prophase, metaphase, anaphase, telophase. They don't necessarily need you to know all of those specific stages and what happens in them, but they do want you to kind of get the general idea. Now, what I'm going to do is I'm going to go through those specifics, because it was my experience that in college it was good to know those. And so you've probably heard a lot of them already, so might as well just go ahead and review them here today as well. So let's just give some general overviews, okay? Meiosis can be broken up into stages, basically 2 stages. There's 2 different cell divisions, okay? The first cell division, which we call meiosis 1, very inventive, very imaginative, right? Sees the homologous chromosomes actually separate from one another. And the resulting daughter cells after the first cell division have 2 haploid daughter cells, which means we've, at this point, after the first cell division, we've taken one set, maybe the set you got from mom, and put it in a separate cell as compared to the other set, which would be put into another cell. So we are taking a diploid cell, and after the first cell division, meiosis 1, we are making 2 haploid daughter cells, and they have replicated chromosomes. This is where I see a lot of the issue come into play, where kids get confused. Okay, they look at diagrams after meiosis 1, and they see all sorts of stuff in there, and they don't know how many chromosomes are in there, because what you actually have are haploid daughter cells with replicated chromosomes. Okay, so it leads to a counting of incorrect chromosome number at this point. Okay, in the second cell division, this is where sister chromatids separate. and we call this meiosis 2. Now, in meiosis 2, the events of meiosis 2 are almost 100% identical to what happens in mitosis, okay, in terms of the events, prophase, metaphase, anaphase, telophase. Okay, it is in the first cell division, meiosis 1, where we see a large amount of differences between mitosis and meiosis in terms of those individual stage events. Okay, so in general, this is a really good, I think this is a really good diagram for you guys to kind of study to get the overall gist of what's happening in meiosis. Okay, we start off with, at the very beginning, we start off with a diploid cell. We know it's diploid because we have one set that came from dad, one set of chromosomes that came from dad, and we have one set of chromosomes that came from mom. One blue, one red equals one set. Okay, so, or pardon me, two sets. Right? We have one set here and one set here. So the 2n number of this, the two set number of this particular cell is 2. Okay, so the diploid number of this cell is 2. All right, so after that we get chromosomes. They replicate, and we see that here. Now each one of these up here has a sister chromatid attached to it. And this is still a diploid cell, okay, because we still have one set that came from dad, one set that came from mom. All that's changed is that we've attached an exact copy to it, okay? Now, after meiosis 1, what we see happen here in this very simplified 2-chromosome system is that we take one of those chromosome sets, okay, dad's chromosome set, we package it into its own cell, and we take mom's chromosome set and we package it into its own cell, okay? So let's take a look at this. We had 2 chromosomes here. We have 1 and we have 2, right? And after the first cell division, I've said that we end up with 2 haploid daughter cells. Let's see if that's true. We know if it's haploid if it's missing the other set, okay, if it's missing half the sets. Okay, so let's look at this cell. This cell has dad's set. Well, we notice that we're missing mom's set. It's nowhere to be seen in this cell, so we can unequivocally say that this cell is haploid because it has half the number of chromosome sets than it did previously. Okay, and this one over here has mom's original set of chromosome, and where's dad's? We don't see dad's in here, so it has half the number of the original sets, so we can say it's a haploid cell. Okay, now after the second cell division, after meiosis 2, all we're doing now is increasing the number of gametes. We still have a haploid set, or a haploid cell, we still have a haploid cell, but now all we've done is separated those sister chromatids into their own separate vessels, and we do that with the ones that we see over here as well. Okay, so what we're going to talk about next is the stuff that you maybe have to loosely pay attention to. I would not spend a massive amount of time memorizing a lot of this. I will point out to you the things that you definitely need to know because they do happen here in meiosis 1. But as you will see those trail off, and as you see me put less emphasis on some of the stages, you should as well. Okay, so the first thing we see is that the cells that are going to undergo meiosis, they go through interphase the same as any other cell, and during interphase they replicate their chromosomes, and so that's actually what's happened in this particular cell. We can't really tell because it just looks like a jumbled mess of chromatin, which it is. But at interphase, we start to see centrosomes form, very similar to what we would have seen in mitosis. Okay, and after interphase, then we enter meiosis 1. And in meiosis 1, we have the exact same phases that we would have seen in mitosis, only they label them prophase 1, metaphase 1, anaphase 1, telophase 1, because they happen in meiosis 1. Okay, so let's go through the events of each. And these are, in prophase 1 is where we see a vast majority of the differences as compared to prophase of mitosis. Okay? Prophase 1 typically occupies about 90% of the time required for meiosis. There's a lot that has to happen here. Chromosomes condense like they normally would in regular prophase of mitosis, but now is where we start to see the differences. Each pair of chromosomes forms what's known as a tetrad, okay? And it's called such because it's a group of 4 chromatids. And basically what happens is when they form these tetrads, they go through a process known as synopsis, okay? And so synopsis is when the homologous chromosomes will find one another. It's kind of crazy that, you know, they could be located anywhere in the nuclear region, and they will actually find each other and match up, okay? So we end up with 23 pairs of homologous chromosomes that ultimately will match up with each other. They will find each other and loosely pair up. And when they do, they form something called a tetrad, because what you'll see is there are basically 4 chromatids kind of grouped together, okay? Also in prophase 1, we see a phenomenon known as crossing over happen, okay? And this only happens in prophase 1. It does not happen anywhere else in meiosis. It does not happen in mitosis. It is unique to prophase 1. And this is where non-sister chromatids will exchange DNA segments. There will be a literal breaking and then reattaching of DNA segments of adjacent non-sister chromatids, okay? And we can see this picture happening here. We can see this here. So at this point here, we see that the centrosomes have formed, and we start to see spindle fibers. That's an event that also happens in mitosis. But what we see happening here is that this maternal chromosome paired up with this paternal chromosome, because they're the same size. They're homologous, right? The same thing we see with these big ones here. The big paternal chromosome paired up with the big maternal chromosome. They went through a process known as synopsis. They came together, okay? And we also see happening here, I can't zoom in real well, but I can't zoom in at all, but if you can zoom into this spot right here, this circle, you'll notice that the legs of the chromosomes, or the sister chromatids, kind of cross over each other. And what they do is they will physically snap the chromosomes off of one another and then exchange arms, okay? And so ultimately what you'll see, and you can't see it totally just yet, but ultimately what you see is a literal exchange of genetic material, okay? And so by this process of crossing over, we can increase the genetic diversity by recombining different types of genes, or different versions of genes called alleles, okay? All right, let's go on to the next phase. Now this is going to be where I start to speed through some of this stuff, because what we end up seeing is that a lot of these phases are very, very similar to what would happen in regular mitosis, okay? So next we have metaphase 1, and in metaphase 1, the microtubules will attach to the kinetochores. That happened in mitosis. And these microtubules will move these homologous pairs, these tetrads, towards the center of the cell, which we call the metaphase plate. All that stuff is kind of the same, other than the fact that now we have homologous pairs of chromosomes kind of linked together, more or less. Okay? In anaphase, this is a little bit different from what happens in mitosis. In anaphase, we have the homologous chromosomes separate. Now, the reason this is different from regular mitosis, because in regular mitosis, during anaphase, we have the sister chromatids separate from one another, okay? And in anaphase 1 of meiosis, we have homologous chromosomes separating from one another, okay? But the movement is the same, okay? These things travel back on the microtubules towards the poles, the exact same kind of, in the exact same manner that you would see happen in mitosis, okay? And then in telophase, we see the nuclear envelopes begin to kind of reform here, okay? Not wholly, not all the way, and we start to see cytokinesis occur. Both of those events are the exact same things that happen in mitosis, okay? So a lot of what we've seen already is exactly what happens in mitosis, with some small differences. The differences we see are in prophase 1. There's some big differences there, so make sure you study that one. And also in anaphase, there is a difference where we have the homologous chromosomes separating as opposed to the sister chromatids. Okay, so there is there is your overview synopsis of what happens in telophase 1, cytokinesis. If you want to learn that, remember it, great. You don't necessarily have to commit that to memory. Okay? Now, in between meiosis 1 and meiosis 2, there is no new chromosome replication. The cell does not enter interphase. It just transitions into meiosis 2. Now, meiosis 2 is almost identical in nature to mitosis. Meiosis 2 is almost identical in nature to mitosis. And as a result, I'm not going to spend hardly any time on it at all. Okay? And so, you know, I can sit here on the diagrams and just overview what's happening. Now, these diagrams are a little bit different because they show you the fate of each one of the daughter cells produced after meiosis 1. So these are the 2 daughter cells produced after meiosis 1, right? Now it shows you what happens to each one during its developmental phase. So prophase 2, the same thing happens. We get the microtubules start to form again. The nuclear envelope's going to completely go away. And in metaphase, we get the attachment of the microtubules to the kinetochores again, and these microtubules move these things to the center of the cell, the metaphase plate. Okay, exact same thing that happens in mitosis. Okay, so if you want to spend time, memorizing these words, you can. It's not a big deal. If you don't, in anaphase, I mentioned that the sister chromatids will separate this time, and so this is again identical to what happens in mitosis, and you see that happening here. The sister chromatids are together in metaphase, and they are now being separated here, and those sister chromatids are going to be moved back towards the poles of the cells. Okay. And then in telophase 2 and cytokinesis, again, almost identical in nature to mitosis. So you get the reformation of the nuclear envelope, you get cytokinesis, which separates the cytoplasm, and ultimately at the end of meiosis, you are looking at 4 daughter cells, which are haploid. Okay, that's important. 4 daughter cells that are haploid. And the chromosomes are unreplicated. Okay, on top of that, each daughter cell should be genetically distinct from the others and also from the original parent cell. That is one of the most important things I can express to you, is that each daughter cell is genetically distinct from the others and also from the parent cell. And the reason for that is because of the crossing over that happened in the very beginning of prophase 1. Okay? There are a couple of other events that did occur that we will talk about that increase this genetic variation that we see in these cells, but we're going to save those to discuss a little bit more in depth separately. Okay? So this is meiosis 2 that you see in front of you, all of the phases in one, and to be honest with you, if I were you, I would just look at this diagram and use that as your study tool for meiosis 2. Okay. All right, so this slide here, and this slide here, and this slide here, I think are going to be great for comparing mitosis to meiosis. Okay. These 3 events right here are unique to meiosis. Okay, and they are 3 of the events that I spent time expressing to you that you should know. And all 3 of these events occur in meiosis 1. I've talked about the synapsis and the crossing over that happens in prophase 1. Okay, and then at the metaphase plate there are paired homologous chromosomes instead of individual replicated chromosomes. And then in anaphase 1, it's the homologous chromosomes that separate instead of sister chromatids. All of those things are things that I expressed to you that you probably should know. And these are the things that the College Board are going to spend a little bit more time on testing you than the actual events of like what happens in cytokinesis 2 or what happens in prophase 2. They want to know this stuff, how these events differentiate meiosis from mitosis. Okay, so this diagram kind of puts it all together, and really what you see here, the big difference lies in the synapsis, because after these homologous chromosomes synapse or come together, you'll notice that they line up distinctly different at the metaphase plate. They're next to each other instead of on top of each other. In regular mitosis, all of the chromosomes, homologous or not, are on top of each other. And just linear on top of each other. And here we see that they're stacked up next to each other. Okay? And ultimately, that's really what results in the differences, is that synapse in the prophase 1. So utilize this. I'm going to probably come back to this diagram in person or live so that we can talk about this diagram a little bit more in general, or a little bit more in detail, and break it down so that I can ask you questions about how many chromosomes are in each cell, and we can distinguish between haploid and diploid. So I think this will be a really good study tool for you guys when you do study. Okay? And then here we have a table that kind of puts some of the major things into perspective for both mitosis and meiosis. I'm not going to spend any time on it other than to tell you that I think it's a good tool for you to study from, because it kind of puts it all in an organized fashion. Okay? Puts all the differences and some of the major things in an organized fashion. Okay, what I do want to do here is I want to play this computer animation for you, because I don't know, again, if you could have access to it. I'm thinking that you can't, but it's a really good overview, and they tell you a lot of the same things that I told you as well. Yes, allow. Okay. Maybe it's not going to work. Okay, it looks like it's broken for me too, guys. So I will try to find a good. I will try to find you a good animation of meiosis, all of the events online, and link it when I provide the video for this as well. Okay, so that leads us to where I'm going to start off the third and final video. I talked not extensively, but I put some emphasis on the fact that the daughter cells are genetically distinct from each other after meiosis has occurred. And so in this final video, when we talk here in section 4, we're going to talk about the importance of this genetic variation in this process of meiosis, and how this genetic variation comes about, and how it plays out when organisms actually partake in the sexual life cycles that yield the baby. Okay? So I will see you in that video.